Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue our lesson today talking about protein synthesis. So far, we've only been talking about what happens when everything goes perfectly right. Today, we're going to change it up and we're going to talk about mutations. This is what can happen when all of this process, the replication, the transcription, and the translation, if any of that doesn't go the way it's supposed to, what you can get is something called a mutation. And we're going to talk about different types of mutations and how they can change your body. Okay, so let's go ahead and hop into it and see what's going on. Okay, so what is a mutation? Well, mutations are changes to genetic information. So it might change up your DNA. Okay, this can be caused by mutagens. Okay, mutagens are things that can cause cancer, and there's plenty of different examples of mutagens. Okay, chemical substances, X-rays, ultraviolet radiation, so even sunlight can cause mutations. Anything that can cause a mutation or might cause cancer is called a mutagen, and these are things that can change your DNA. Okay. Mutations can happen on large or small scales. When we're talking about large-scale mutations, we're talking about chromosomes. So when chromosomes have mutations, entire pieces of the chromosome can break off and attach to different chromosomes or get deleted or lost. And so those are very large-scale because remember, chromosomes, it's a lot of DNA packed very tightly. Okay? You can also have small-scale mutations, and this is mutations on the nucleotide level. So instead of losing an entire half of a chromosome, we might just change one nitrogen base for another nitrogen base. Sometimes those can have effects and sometimes they don't have any effects. So we're going to talk about some examples of large-scale mutations first. The reason that I like learning about these is that the name of the mutation is kind of a hint to what's going on. So one type of large-scale mutation is known as deletion. In a deletion mutation, part of the chromosome gets deleted. So just like the delete button on your computer erases whatever you just typed, in a deletion mutation, this is when some of the chromosome gets deleted or lost. Okay? Another type of mutation is known as duplication. If you were to duplicate something, that means you make an extra copy of it, right? And so in a duplication mutation, part of the chromosome gets duplicated. So the chromosome's like bigger than it's supposed to be. Another example of a mutation is called an inversion. In an inversion, two parts of the chromosome gets reversed. Okay, and we'll look at a picture of these in just a second. Another type of mutation is called an insertion. In an insertion, one part of the chromosome gets inserted into a different chromosome. So this is when it would break off of one chromosome, move over to the other one, okay? And then another type of mutation is known as translocation, and this is when parts of two different chromosomes change location. So let's look at some pictures of these and see what we can figure out what's going on here, okay? So look really closely at this first example. This is known as a deletion mutation. On the left, we have the full chromosome, and then you can see that a little bit of it got lost or deleted. And so on the right next to it there, that chromosome is a little shorter because we've deleted part of it, okay? And this could be really bad because that might have some genes that were really important that we really needed. Okay, what are some other examples of these mutations? Let's look at this one. This is called duplication. Here almost the reverse of the deletion is happening. On the left, the chromosome is smaller, and then once we duplicate a portion of it, you can see that right next to it we have two parts of those chromosomes stacked on top of each other, and so the chromosome gets a little larger there. Okay? Another one is known as inversion. This one isn't really changing the size of the chromosome, but if you follow the arrows there, you can see that that portion of the chromosome got turned upside down. Okay? Here are some other examples. This one is called insertion. So in insertion, what we do is we take one piece of one chromosome and put it into a different chromosome. So here we have the yellow chromosome and the gray chromosome right next to it. What we're going to do is part of the yellow chromosome gets moved into the gray chromosome. And so now the yellow chromosome is a little smaller and the gray chromosome is a little larger. Okay? This one is known as translocation. This is when pieces of two different chromosomes switch spots. So you can see we started with a totally green chromosome on the left and then a totally red chromosome on the right. After this translocations happen, we have a green chromosome with a little red and a red chromosome with a little green. So those two pieces of chromosome have swapped places. So these are examples of large-scale mutations. What I wanted to talk about next is smaller-scale mutations, and we call these point mutations. These are mutations on a much smaller level. Sometimes just a single nucleotide base is affected instead of large portions of a chromosome. Here are some examples of point mutations. So we can have something called a substitution. This is when one nucleotide gets replaced with a different base. Again, to me, the name kind of tells you what's going on. Imagine you came into class one day and instead of the teacher that you're used to seeing, you had a substitute, right? That substitute, they're the teacher, they're doing the same job, but it's just a different person. That's very similar as to what's going on here. It's when one nucleotide gets replaced with a different base. Okay, this may or may not have an effect. In the best case scenario, this would code for the same amino acid and you wouldn't even know that a mutation has occurred. These next two can be a lot worse. So one of these is called an insertion. In an insertion mutation, an extra nucleotide gets added. And in deletion, this is when a nucleotide base gets deleted. Okay, and I'll talk about why these are a lot worse here in just a second. Okay, so let's talk about which of these point mutations are really, really bad and which one of these may not be as bad. So some mutations are worse than others. From smallest change to largest change, so the ones at the top up here are going to have little to no change, and then the two at the bottom can have large scale changes. The best type of mutation to have is called a silent mutation. And a silent mutation, a mutation that codes for the same amino acid. We learned last time that a lot of those codons are coding for the same amino acid, especially if you're changing the third base in the codon. If you change the third base in the codon, you might not even be coding for a different amino acid, so there would really be no change here. And if there's no change, we call it a silent mutation. A different type of mutation is known as a missense mutation. What's going on here is that a mutation that codes for a different amino acid, and this may or may not affect the protein being made. If you change the first base in a codon, you're more likely to make this type of mutation because that first base in the codon is really, really important. All right, and then these two are a lot worse. We can have something called a nonsense mutation. And a nonsense mutation, what's going on here is that this is a mutation which causes for a premature stop codon, and that results in an incomplete protein. So last time we learned that ribosomes know where to stop and start the protein's synthesis process by using what are known as stop and start codons. Well, imagine a nonsense mutation would be like if you put a period in the middle of a sentence. It just kind of ends abruptly and it's kind of confusing, right? So in the same way, if you were to change an amino acid and now that amino acid is a stop codon, you're not going to build the full protein because it stopped in the middle. It's like putting a period in the middle of a sentence. The worst type of point mutation is known as a frame shift mutation. These are caused by insertions and deletions. What's going on here is that the insertions and deletions are going to change the codon reading sequence. Remember that we read codons in sets of 3s. So if you add or delete one nitrogen base, all of the codons after that are wrong. So let's look at this real quick and then I'll tell you more why those frame shift mutations are so bad. So up at the top, we've got our DNA molecule. What we're doing here is that transcription process. You can see that the DNA is opened up and we're making an mRNA molecule, okay? The first one underneath that is just a normal one. This is when everything goes correctly and we found all the complementary base pairs correct. What can happen though is we can make something called a missense mutation. This is when you have changed one nitrogen base for a different nitrogen base. So this could be just putting a C where a T needs to be. This may or may not cause a change in amino acids and it might not even change the protein that you're making. Okay, the worst ones are these frame shift mutations. These are caused by insertions and deletions, and what's going on here is it changes that 3 codon sequence reading pattern. Now all the codons after it are going to be wrong, and I'll show you a little in more detail here on the next slide. And then the last one there is a nonsense mutation. That's when you are coding for a stop codon early. That's like when you put a period in the middle of the sentence. The ribosome is going to start building that protein, and then it stops before the protein's done being made. All right, let's look at those point shift. Let's look at some of these different point mutations real quick. So over on the left, you can see that's our original. We have AUG, CAG, UCG, CAG. And then you can see that those are the amino acids being coded for down there. That AUG is our start codon, and that is always methionine. So that's what the MET stands for. And then you've got 3 amino acids after that. Okay, well, let's look at the one below it. This would be a missense mutation. This is when you change one amino acid for a different one. So look at that G that's pink. In the original, that G was actually a C. So what would happen if we changed a C for a G? Well, now look at the amino acids at the bottom. They're not the same. We have methionine, but where that mutation has occurred, instead of the ALA, which is alanine, now we have TRP, which is tryptophan. So now we're coding for a different amino acid, and this is going to make the protein different because it's being made with different amino acids. Okay? On the right, at the top right corner, that is a nonsense mutation. This introduces a stop codon where there shouldn't be one. Here, that U that is pink, in the original DNA sequence, that was a C. But if it gets coded as a U instead, now we've got a stop codon there where there isn't supposed to be one. And then the last one in the bottom right corner, that is a silent mutation. This is when no change in amino acid has occurred. You can see that there's an A there, that's pink. Originally that was a G, but CAG and CAA code for the same amino acid, so it doesn't really have a change at all. So that's why it's called silent. Okay, let's look at the frame shift mutation and let's figure out why these are so bad. So what I really need you to know is, remember, when we read mRNA, we read it 3 bases at a time, okay? 3 bases is known as a codon, and that's almost like making a word in a sentence. So let's look at this wild type. This is the original. This is what it's supposed to be. You can see that there's a lot of letters there, but if we break it up into groups of 3, breaking it up into group of 3s is like reading it as a codon. We can read this sentence very clearly. It says, the old man and the sea, right? These are 6 codons, and we know what each codon is supposed to represent, okay? Well, look at the mutations at the bottom. The first one is if we were to add an extra nitrogen base. So this would be like an insertion. Now, we've only added 1 letter, but try reading that sentence now. It says the, and then XOL, DMA, NAN. You see how now all of the words after that insertion is wrong? Is because we're still reading it in groups of 3, but the frame that we're reading them in has shifted. And so now all of the codons are incorrect. The same thing at the bottom there, if we were to just delete a nitrogen base, so here we're deleting the O, the first word is still correct, the, but then every word after that, every codon after that is wrong because now we have shifted the frame at which we're reading the codons in. So I hope that kind of clears up the frame shift and the different types of mutations that you can see. Thank y'all so much for following along with me. Your teacher has some other work for you to practice on, so make sure to give that your best effort, and I hope everybody has a great day.